talking about the important entity, that is the diarrhea. Okay, now talking about the diarrhea. Acute diarrhea, that is classified as non-inflammatory, that is watery or non-bloody, and one is inflammatory or also termed as bloody. Fine. So acute diarrhea is classified as non-inflammatory or inflammatory in total. What is the meaning of this particular thing? The meaning of this particular thing is this, that the non-inflammatory means that there is no inflammation. It is due to the toxin mechanism and all that which have happened up, like in the case of Vibrio, a typical cause. Whereas the inflammatory one happens to be in whom, like in the Salmonella, Shigella or Campylobacter. That particular thing is termed as what? Inflammatory stuff. That is the, and in that inflammation, like invasiveness can also come, and that can turn out to be what? Bloody also. Fine. And bloody diarrhea, you already know, is termed as dysentery. Okay. For example, the bloody diarrhea mainly done by whom? Shigella, and that is termed as what? Bacillary dysentery. And that describes the important features of watery and the bloody diarrhea. Okay, so in total what I want to tell you here is that the acute diarrhea can be of non-inflammatory cause or inflammatory one. Non-inflammatory is watery or non-bloody. Inflammatory is bloody, and bloody diarrhea, the most important cause is Shigella to happen up. Okay, and to whom you call the diarrhea and all. The diarrhea is relative. It must be calibrated against the patient's normal bowel movements. Fine. And it is usually considered to be greater than 3 to 5 bowel movements per day. If it is so, then it is termed as what? Diarrhea. And most of the infectious agents that cause diarrhea, they act where? They act at the small intestine. Where they act? They act to the small intestine, where the majority of the fluid get absorbed. Or the colon also they can act upon, but majority where they act on the small intestine. So there is a table in the Harrison which you can look out, and that's a wonderful table, and I will show you that particular table. This is that particular table, fine, which classifies these diarrheal agents into non-inflammatory, inflammatory, and the penetrating one. Okay, so first of all, if we talk about the non-inflammatory one, okay, see the location. This is very important. Proximal small bowel, that is the location of it. Okay, what is the illness happened? That is the watery diarrhea, because this is non-inflammatory. Okay, what is the stool finding? This is also important. There is no fecal leukocytes, guys. Okay, there is no fecal leukocytes here, and fecal lactoferrin is not increased. Why? Because leukocytes are not there. Got my point? So this is non-inflammatory one. Now, if somebody asks you that which particular bacteria they are involved up in causing this, then this is the various bacteria which you need to focus upon. The number one is Vibrio cholerae, I've just told you. Then you have this E. coli one. Which one? Enteropathogenic, enterotoxigenic, or enteroaggregative. These three are there. One is pathogenic, one is toxigenic, and one is aggregative. These are the ones which are there in the non-inflammatory E. coli. Okay, they cause non-inflammatory diarrhea. Which one? Pathogenic, which is common in the pediatric age group. Toxigenic, traveler's diarrhea. Aggregative, persistent type of diarrhea. Fine. Then you have this Clostridium perfringens. This also relates with the diarrhea, and that is non-inflammatory one. Bacillus cereus, diarrheal one. Then you have the Staph aureus. This is also non-inflammatory. Then Aeromonas and Plesiomonas. These are also important ones which associates with the non-inflammatory form of diarrhea. These are the bacteria which are involved. But if somebody asks you regarding the viruses, in the viruses what you have the rotavirus, you have the norovirus, and certain adenoviruses also. Okay, they associates with non-inflammatory form. Remember, rota is a non-inflammatory form of diarrhea. Fine. Regarding the parasites, Giardia lamblia, Cryptosporidium, Cyclospora, Cystoisospora, you know that these are what? These are coccidian parasites. And a separate one, Microsporidia, they all are what? Non-inflammatory form of diarrhea. And they happen up in whom? They happen in the HIV positive individuals. They have a strong correlation with the HIV positivity, and they land up the individual with what? Non-inflammatory form of diarrhea. Got my point? So all of these points are very important. These bacteria names, these viruses, and these parasites, they relate up with whom? Non-inflammatory form of diarrhea. Now talking about the inflammatory form of diarrhea, and this inflammatory form of diarrhea associates with invasion or cytotoxin. Okay, and what is the location, guys? See the location. Just now we have done na, that's small bowel is involved up. Here also, like distal small bowel, colon, that is involved up, okay, in the inflammatory invasion or cytotoxin mediated it can be. Okay, why it is saying so? Because of this only the blood and all will come. That's why it is termed as bloody diarrhea. Now predominantly the dysenteric episodes, that is the bloody diarrhea ones, they are done by whom? Shigella. Okay, a close Campylobacter. Then as the name suggests, blood enterohemorrhagic, enteroinvasive, which resembles shigellosis, and Vibrio parahemolyticus, which is a halophilic Vibrio. They associates with dysentery like features in this inflammatory form of diarrhea. Okay, now but there are some other forms of diarrhea also in which blood does not come, but they come under which category? Inflammatory one only. Let's have the name of them also. Salmonella. Shigella bloody, but Salmonella is not bloody diarrhea. Okay, Yersinia enterocolitica, Listeria monocytogenes, Clostridium difficile. Clostridium difficile CDAD, you know that. Okay, Aeromonas and Plesiomonas are here also. Okay, Aeromonas and Plesiomonas means they can cause both inflammatory as well as non-inflammatory one. Okay, so that is also to be remembered. Now coming back to this, if we talk about in the inflammatory one regarding the parasites, fine. The parasites which causes predominantly dysentery is, two names immediately come to your mind. One is Entamoeba histolytica and one is Balantidium coli. Okay, Balantidium coli, the largest intestinal protozoa which does inflammatory form of diarrhea, but mostly that is bloody. Okay, and Entamoeba histolytica, that is also related with the bloody dysentery. Got my point? Then coming to the stool findings of the inflammatory form of diarrhea. Stool findings of the inflammatory form of diarrhea, and this is a stool finding. What are those stool findings? The fecal pus cells, obviously that will be observed of fecal leukocytes, that is the polymorphonuclear leukocytes, they are increased of, that's why we are saying it is inflammatory. Then the fecal lactoferrin, they are increased of in this particular one because leukocytes are increased, so indirectly the lactoferrin will also increase. Now coming to the third variety, that is termed as penetrating. In the penetrating, the location is distal small bowel. Okay, so basically small bowel is involved up in the diarrheal episodes mainly, and the illness named is enteric fever. In this penetrating one, it penetrates the M cells, you know that na, mucosal cells and all, and the stool finding will have what? Mononuclear leukocytes. Fine, fecal mononuclear leukocytes, they are observed up, they are increased in this particular one, and obviously you know enteric fever is done by whom? Salmonella typhi, and typhoid like illness is done by Yersinia enterocolitica, which also produces what? A super antigen. Okay, so all this particular table is beautifully made. It is telling up regarding the non-inflammatory ones also, it is telling regarding the inflammatory ones also, and in this particular one you are seeing up penetrating also. So basic difference what is coming out of all this is that in the non-inflammatory one there is no fecal leukocytes, no fecal lactoferrin is measured off, but in the case of invasion or cytotoxin mediated inflammatory one, in that you have the fecal lactoferrin also, fecal leukocytes are also seen, but this is divided into two things. One is the hemorrhagic one, that is the bloody one, and one is the inflammatory one which does not have the blood. The bloody one, the predominantly number one is Shigella, Campylobacter is a closed one, hemorrhagic is coming up in this, enterohemorrhagic, invasive is there, and Vibrio parahemolyticus, that is a halophilic one which is in the inflammatory bloody. Whereas in the parasites, if you go for Entamoeba histolytica and the Balantidium coli, and this is predominantly the inflammatory diarrhea, means the blood is not there. The close brother of Shigella, Salmonella, does not cause bloody stuff. It does not love blood. Yersinia enterocolitica, Listeria monocytogenes, Clostridium difficile, that is CDAD. Okay, Aeromonas, Plesiomonas, they are common to both inflammatory as well as non-inflammatory one. And in the penetrating, you have the two examples of Salmonella typhi and Yersinia enterocolitica, which are there in the inflammatory also. Okay, so I suppose you got this particular point. Okay, now in this particular diarrheal episodes only, we should like talk about the food poisoning infectious agents also. Okay, we should talk about the food poisoning infectious agents also, that what are the various agents which causes food poisoning and all, because that is also a favorite question of the examiner. Another chart from the Harrison. Okay, there is another chart from the Harrison, this particular one, the various infectious agents of the food poisoning. Now in this, if the time given up in the question is 1 to 6 hours, okay, which all organism they associate up? The first one is Staph aureus, then you have Bacillus cereus, and then you have this Clostridium botulinum. Okay, you know na, Clostridium botulinum, this also associates with the like vomiting, nausea and all that stuff. Okay, obviously diarrhea also. The Bacillus cereus diarrheal is also there, emetic is also there, and Staph aureus also associates with all these things, nausea, vomiting and diarrhea. So all of them, they associate with these particular features, nausea, vomiting and diarrhea. And the incubation period for them is 1 to 6 hours. And what is coming in them? Staph aureus, Bacillus cereus and Clostridium botulinum. Okay, Clostridium botulinum also comes in 1 to 6 hours. Remember this. Okay, now what particular thing you have to catch up on in the exam is that what they have eaten. So in the case of Staph aureus, you need to look out that what they have eaten up. So this is the thing what they have eaten up. What is that? A ham, poultry product, okay, potato or a egg salad, fine, mayonnaise, pastries, your favorite things. Okay, these, if they are given up in the question, you should think for whom? Staph aureus. If the incubation period is given what? 1 to 6 hours. First thing. Then Bacillus cereus, emetic, strongly associates with whom? Fried rice. Which type of fried rice? Reheated fried rice. Heat stable toxin is involved. What is the mechanism of action? Increase cyclic GMP, guanosine monophosphate. And Clostridium botulinum, you already know, that associates with what? Canned food. Okay, canned food associates with the Clostridium botulinum, this particular nausea, vomiting and diarrhea. And one particular thing I want to tell you, that the canned food poisoning is not having this particular feature. This is wrongly written here. This is mainly having the constipation. Okay, so this is wrongly being written here, that it lands up with the diarrhea. But yes, it is not a 100% rule that it will land up with the constipation, but majority of them, they land up with the constipation in this particular Clostridium botulinum verb, and because anticholinergic action is there. So canned food poisoning associates with constipation rather than the diarrhea. This also needs to be focused upon, and this is a question also. But yes, diarrhea can happen. Now 8 to 16 hours. The next one, if we take the timing, that is 8 to 16 hours. 8 to 16 hours, you have this Clostridium perfringens, fine. And what are the features? Features are abdominal cramps, diarrhea, and the vomiting is rare here. The vomiting is rare. And see the things which is being eaten up. Contaminated meat like beef, poultry products, legumes and gravies. That is associated with Clostridium perfringens. Okay. Bacillus cereus. Bacillus cereus, this we are talking about the diarrheal one. We are talking about the diarrheal one, and even in the diarrheal also vomiting can happen. So diarrheal is the main one along with the abdominal cramps. And what they have eaten up here? Obviously contaminated meat and milk. So meats, vegetables, dried beans, cereals along with the milk and all. Okay. So that is associated with whom? That is associated with Bacillus cereus diarrheal. Now coming to the another entity, that is the Vibrio cholerae, watery diarrhea and the shellfish and the water. If they are contaminated, ingested up, that will lead to the infection of cholera. Enterotoxigenic E. coli, that associates with watery diarrhea, non-inflammatory one, associates with whom? Associates with salads, cheese, meat and water. Okay, and this leads to what? Traveler's diarrhea. Okay, I will discuss that in the later slide. Then you have this enterohemorrhagic E. coli, that is there, that is the bloody diarrhea as the name suggests, and this associates with the meat products like ground beef, then raw milk, raw vegetables, and apple juice. And how you can remember that apple juice is involved? Red, both are red. Okay, then you have this salmonella. This associates with inflammatory form of diarrhea. Fecal leukocytes will be seen, and this one associates with beef, poultry products, eggs, and dairy products. And obviously these are non-salmonella typhi ones, typhimurium enteritis and all that. Campylobacter jejuni, this is also associated with inflammatory diarrhea. Okay, and this particular one associates with this particular poultry and raw milk. Poultry and raw milk, Campylobacter also goes hand in hand. I've told you na, that whenever they talk about the poultry products and all, you should think for Salmonella, Campylobacter, these two names for sure. Okay, so here also they are talking about the both. Shigella 1 associates with the dysentery, bloody diarrhea, and the causal agents is potato or egg salad, lettuce or raw vegetables. Fine. And Vibrio parahemolyticus also associated with dysentery like seafood poisoning, and mollusk and crustaceans are involved up in that seafood. Okay, so slight bit because of a slight shift in the table, this particular confusion can happen to you, but I have marked up with the arrows that which one associates with what. So 1 to 6 hours, these three names, 8 to 16 hours, these two names, perfringens and bacillus cereus, and more than 16 hours, these all names, they associate with the food poisoning. Are definite question. Okay, now talking about another definite question, that is the traveler's diarrhea. And what is traveler's diarrhea? If an American or a British fellow comes to India, drinks our water, okay, definitely will land up with what? Diarrhea. And this is what is termed as traveler's diarrhea. Okay, and if that particular individual from America or UK like have our pani puris and all, then he will land up in the nearby hospital very soon with a severe diarrhea. Okay, and why does it does not cause anything to the Indians? Why the COVID-19 is becoming a what do you say, joke for the Indians? Because we already have so much of viruses, they are interfering with each other, that COVID is not having much of the relevance for the Indians. Okay, and this is the reason that it is not spreading like a wildfire as it is spreading in the UK, US and all Australia and all that, because we are already struggling with millions of viruses. Then COVID does not have that place in our life, okay, or in our gut, so that it can cause something to us. Okay, so as such, jokes apart, what are the various agents which cause the traveler's diarrhea? So for remembrance of that particular one, that you can remember a mnemonic which is made by one of my students only, that is VIP ECG scanner. Okay, this particular mnemonic will help you to remember the various causes of traveler's diarrhea. A definite question of INCT in which they can ask all the following are doing the traveler's diarrhea except. So what is this VI, guys? So VI stands for Vibrio. So Vibrio cholerae can cause traveler's diarrhea. Okay, this P stands for Plesiomonas. Okay, this particular P stands for Plesiomonas, as you can see this name in the front also. Then this E is Entamoeba histolytica. Okay, as you can see down, Entamoeba histolytica. C is Cryptosporidium. C is what? Cryptosporidium. Fine. And this G is Giardia. This G is Giardia. Okay, I'm removing it because scanner I have to unfold. So all these three parasites, they lead to traveler's diarrhea. Okay, now talking about those scanner. Okay, VIP ECG scanner was the mnemonic na. So S is what? That is the Salmonella and Shigella. These are there. They also associates with the traveler's diarrhea. C is Campylobacter. C is Campylobacter. A is Aeromonas. A is Aeromonas. N is Norovirus. Okay, N is Norovirus, and this E is ETEC and EAEC. Okay, Enteroaggregative E. coli, Enterotoxigenic E. coli, and this R is rotavirus. This R is rotavirus. And can you tell me guys, which is the single most important cause? This E is the most important. ETEC is the number one cause, as you can see in the first line. This is the single most important agent, and these all which is there in our mnemonic. What is the mnemonic? VIP ECG scanner. Okay, and that includes up all of these lists and a definite question which can come in your exam. So I would say definitely you should go for it. Regarding the neuro, I want to tell you one more thing which can come, that is it associates with cruise. Okay, cruise, if you're going on cruise and all, and they have told up a story behind the cruise, so neuro you have to like predict of, you have to suspect the neuro infection. Rota, if it is talking about the children's and all, that is there, fine. So all important ones, or they can make a story of cryptosporidium, that is there in that particular list also. That is the coccidium parasites which cause diarrhea in the HIV positive and all. The same story is for cyclospora. Okay, so all of them, they leads to what? All of them, they leads to traveler's diarrhea. And what is the mnemonic for this? VIP ECG scanner. And what is there in the ECG? In the ECG, they are the parasites. VI stands for Vibrio and P stands for Plesiomonas. And this A is Aeromonas. I suppose this particular thing helps you. Fine. Now, besides all this stuff, I want to tell you what is our microbiological significance here. That what happens in the Vibrio cholerae, what the features of the Shigella, what the features of this Salmonella and all that which you get up. Okay. Now, can you tell me, like I will be asking you here. So Vibrio cholerae leads to inflammatory or non-inflammatory form of diarrhea. So obviously it is watery diarrhea. So this is non-inflammatory form of diarrhea. Does it lead to traveler's diarrhea, guys? It leads to traveler's diarrhea. Okay. VIP ECG scanner. Then, what is the important feature of Vibrio? It associates with darting motility. First thing. Then the comma-shaped gram-negative bacilli in the culture smear, that is observed. Okay. That will be observed for sure. Okay. Then it is catalase and oxidase positive, a very important reaction to be remembered. Catalase and oxidase positive, this is, they will show the darting motility, that will be shown up in the question. And when you use this best media for this, which one? TCBS. And what is the full form of TCBS? Thiosulfate, citrate, bile salt, sucrose. This particular name, if they talk about, then which type of sucrose fermenting colonies colored are there? Yellow colored. Okay. So on a green media, if you are changing anything changing to yellow, that means they are talking about the TCBS and they are talking about the Vibrio cholerae. Because of the sucrose fermentation, it will change the color from what? Green to yellow. First thing. Then you have this test also, eye cut, which is not very essential for the Vibrio diagnosis, because other than that also number of things are there to do. Can you tell me some? String test is there. Okay. There is a fish in stream appearance seen in this. Okay, the important important transport media is what? VR and Carib layer. The best media is what? TCBS here. Okay, so these also you need to focus upon. Agglutinates with the Vibrio cholerae oven antisera. Another important question. That if you want to diagnosis of oven Vibrios, then you have to use straight away the antibodies antisera, that is the antisera, and with that they will agglutinate. One thing. Okay, and if you want to subtype also, then you have available Ogawa antisera, which is the most common pattern observed up in this oven Vibrios. Oven Vibrios are the two types, classical and Altor. So this is regarding the Vibrio cholerae, the questions which can come in and around that. No need to focus upon the eye-cut test, that will not be asked. Fine. Then, talking about the Shigella, can you tell me it is inflammatory or non-inflammatory? This is inflammatory. Which one? Bloody or just inflammatory? It is bloody. That is dysentery. Now, regarding this particular one, gram-negative bacilli, and this is the blunder which the book have done, because this is non-motile. Okay, this is the non-motile one. Remember this particular point here, that the Shigella, it causes dysentery and it is a gram-negative bacilli which is non-motile. Okay, this is the most important feature of it. Then it is catalase positive. Why? Because it is an Enterobacteriaceae member, gram catalase positive. Which one is catalase negative in the Shigella? Shigella dysentery type 1. Another important question. And it is oxidase negative. Another important hint which you will get in this question. Then you have this McConkey agar or DCA. Okay, what is DCA? Deoxycholate agar or McConkey agar. Which type of colonies they come? Non-lactose fermenting translucent colonies. This is the question. Okay, non-lactose fermenting translucent colonies are observed up in this particular McConkey agar or DCA, deoxycholate agar. Then in the XLD for the Shigella, you will get the red colonies without the black center, which is present in whom? Salmonella. But it will not be there. And XLD stands for what? Xylose lysine deoxycholate. Okay, eye-cut test is not important here also. Okay, and this agglutinates with Shigella polyvalent antisera. So that is one of the ways by which you can do this lab diagnosis. You can use this polyvalent sera, and specific monovalent antisera also available up if you are going subspecies like dysentery type 1 and all that stuff. Okay, so this is regarding the Shigella. Now talking about the Salmonella, and Salmonella is again inflammatory. The fecal leukocytes will be observed. Then this is a gram-negative bacilli which is motile. Okay, catalase positive, oxidase negative. Catalase positive, oxidase negative. This one also, because this is Enterobacteriaceae member. On the McConkey agar, this one also produces NLF colonies. On the DCA, again NLF colonies with black center. Okay. In the XLD, the red colonies with black center. This was one of the questions of PGI Chandigarh. The red colonies with black center on XLD is seen in. Answer is Salmonella. Then eye-cut test is important, not important here also. And this also agglutinates with what? Salmonella poly-O antisera, and serotype specific also is available in the market. So we have discussed up three important ones: Vibrio cholerae, Shigella and Salmonella. Then talking about the viral agents. In the viral agents, you have these particular agents: Rota, Noro, Cruz. Okay, adenoviruses 4041, they associate with diarrhea, and the Rota in the children. And how does you do the lab diagnosis? You will take the stool specimen and you can go for the electron microscopy. For the viruses, electron microscope is used to see up those. Okay, and this particular one, the detection of the viral antigen by the ELISA, that can also be done, and detection of nucleic acid, RNA or DNA by the PCR in the stool specimen is the best. Okay, that is the best to go for the virus. This also needs to be focused upon. Now talking about some intestinal parasites, important one. Entamoeba histolytica. Can you tell me what it does? Dysentery. Inflammatory, non-inflammatory. Obviously dysentery is inflammatory only. And what you observe up? Trophozoites or quadrinucleate cyst in the stool sample. And these trophozoites show up a particular entity that is termed as what? That is termed as erythrophagocytosis. Okay, and there is a detection of specific antigens like lectin or specific genes in the stool. That can also be done for this particular Entamoeba histolytica. Lectin. Giardia. Giardia leads to fatty diarrhea, foul smelling diarrhea. Okay, and the trophozoites is a teardrop shape binucleate one with the exostyle. And there is 4 pairs of flagella, and which particular motility? Falling leaf like. Fine. And along with that you have this quadrinucleate cyst, sometimes bi also, with a central exostyle. Okay, remember regarding the Giardia. Steatorrhea happens here. Drug of choice, metronidazole. Trichuris trichiura, that is leading to the dysentery, again bloody stuff. Barrel shaped egg with mucus plugs, asked question of all India. Okay, bile stained it is, and it is having a barrel shape with mucus plugs. Enterobius vermicularis, pinworm. Nocturnal anal pruritus is done by this particular fellow, and it is having a plano-concave egg. This FMG exam question, it contains tadpole-like larvae, and it comes in the list of whom? Non-bile stained stuff. Okay, Scarus lumbricoides, this associates with whom? Malabsorption. Okay, and this is controversial. Some say it does not do, some says it does. Okay, so this is controversial, and it is having the fertilized egg, unfertilized egg, that is observed up in the stool. Okay, how to identify? Unfertilized is elongated. Okay, this is elongated, whereas this is proper, round and oval, and the fertilized one floats in the saturated salt solution. Okay, this is another PGI question. Then unfertilized dips down. Talking about the hookworm is also termed as Ankylostomata duodenale, associates with what? Diarrhea and anemia. Even treachery is also associated with anemia, and the egg of this is having what? 4 blastomeres. Typically 4 blastomeres is there, and there is a clear space. Important to remember is it is also non-bile stain. Then you have the strongyloids, stercoralis, which also associates with the diarrhea. And what you detect up in the stool sample, another INCT question, that you able to find out what? Rhabditiform larvae in the stool microscopy. In the case of what? Strongyloids. Okay. So these are the various intestinal parasites also we have talked about and their features. So you have to just see up this particular tables again and again, then only you could able to manage up most of the things. Okay guys. So these are the important entity which you need to focus upon in the case of diarrhea. Okay, and if you get down on these tables also properly, your diarrheal particular question which is coming up in the exam, in the lab diagnosis also and all, lot of things will be there with you to write up. Okay, and this is definitely a must know for various exams. Okay, and according to the various agents and all, I have defined up that this is traveler's diarrhea agents. These are the various ones which does the food poisoning, another very important question, and these are the ones which is telling up regarding the non-inflammatory, inflammatory and penetrating form of diarrhea.